Yeah, hi everyone. Today I want to speak about the area in front of our laboratory, a very beautiful area, a recreation area. This picture was taken during the winter, and you can see there's been some mist floating down the river, and we get this now and again. And I really want to highlight today the richness of the local marine fauna, how it contributes to a high biodiversity in the zooplankton, and what a important scientific resource these inshore waters are. Most of the sampling from MBA and also from PML is out at station L4, we call it, which is 12 miles from this point here. That's a very reasonable place to have it rather than doing it in the Sound, because the Sound is very dynamic, changing with all the tides. It just changes far too much. If you want to do a long-term survey, you need a far more settled spot. And this is typical of most of the long-term surveys that are done around the world. They have it well out of the sea, out of the influence of the land. I don't know if PML do any research within the Sound or the university. I know we used to do quite a bit in the MBA in times of heroine. That is like mine, Marie Limbour, who did a lot of work on the early stages of various organisms, like some of the crabs, etc. And she would keep them and culture them and draw them as she went along. You've got to to do these sort of studies, you've really got to start off with the adults and the eggs to be sure that what you're drawing belongs to that particular species. But since then, there are other people been working, but I don't think we do very much locally ourselves. I'm going to include a larger area than just the Sound, because all the area around about contributes to the biodiversity in the Sound. And it's a very complex area. Starting off up past the Lyra Bridge, we've got the Plym coming in, the estuary, muddy estuary at low tide. Then we've got a large area going off called Who Lake, which is quite an interesting place to have a walk around. With Sutton Pool going into the Barbican, Millby Docks and the inner docks there. All these have got loads of marinas in them, which also give a larger area for benthic animals to settle to increase the biodiversity. Then go up the river past to Admiral Tard's Stonehouse Creek again, and more marinas there. Millbrook Lake, a very large inlet off to the left. St. John's Lake, massive. Then further on up the river, which is not on this old MBA map, there are a large inlet going into St. Germans and the River Liner. And when you stand further up the river here and look out across some of the water in the Tamar estuary here, it looks like the Mississippi. It's really a very large area of water. When the tide goes out, it leaves large mud banks. So obviously great places for a lot of organisms to grow and live. So it's the features of the Sound. For a start, it's very sheltered. You can see the breakwater out here, which keeps some of the roughest weather away. We've got, as I said already, we've got many inlets, bays, marinas, muddy bottoms, sandy bottoms, rocky bottoms, different depths. So a terrific number of habitats to suit all sorts of different organisms. There's large freshwater inflows from the Tamar Liner and the Tavy, and also from the other side of the Hoe from the Plym. And these, of course, carry lots of organic particles down because of the strong tidal mixing, and lots of nutrients from farmland runoff. This strong tidal mixing, along with various wind events that we get storms, will resuspend the particles. So there's tons of material in the water to feed the bottom living, the benthos species, filter feeding species, and also the plankton, which may also be filtering the water out. We've got, at certain times of the year, quite a lot of oceanic inflow, currents coming up from the Mediterranean area, bringing a lot of more exotic species. We've got a very mild climate. I see people in swimming here every morning, so it must be quite warm. And all this encourages high benthic and pelagic biodiversity in the plankton, and also year-round production inshore. Here in the inshore waters, if you look at the plankton, you'll find them. A lot of them are just reproducing all year round, carrying eggs, young stages, really going pretty fast at it. Whereas if you go much, it'll also happen at L4, but you go a bit further out towards the Celtic Sea, and it's just a desert during the winter. So we know quite a lot about what is in the Sound and the area around about Plymouth from the Plymouth Marine Fauna, a publication that many of you will be familiar with. It was last published in 1957, which was quite a long time ago. And it details all the organisms and all the fauna. It doesn't do the flora, the fauna, the protozoans and animals which have been found locally. And it gives them a list, some information about when they were found, where they were found. So if you were interested in a particular species, you got a chance of going into this book and then going out and finding it yourself. I was interested to see how many species there had been reported locally. Unfortunately, they don't give a total in the book. But in the past, I've noticed things like, I've had to, for talks and things, I've checked up on certain things like fish. You could 175 species I've recorded, probably more than that now. And mollusks, 375 species. So it's indicating a very high biodiversity. So I did a rough count by estimating 8 per page and multiplied it up by the number of pages which are in the fauna, and came up with a number of greater than 3,400, and that's conservative species which have been recorded. So this was, when the MBA was cited here, this was a good place to put us. And subsequently, the Plymouth Marine Laboratory coming here, and it's been good for the university with marine science courses to be in this place as well. So a lot of these, the benthic species have got planktonic stages. They need these stages for dispersion, where the young stages go into the plankton, feed and grow, and then when they become too heavy to swim, they settle out and take up an adult existence. So this promotes a great biodiversity in the plankton, all these young stages, all these species. And the species range is boosted by seasonal oceanic visitors. And in fact, here we have most of the northern European shelf zooplankton species, just about everything in Northern Europe where it is found here. And if you were to add in things which are not there, like the algae, all the phytoplankton, all the seaweeds, etc., which we get in the Sound, fungi, which people in the lab work now in, myofauna, which is a relatively recent science, which is the organisms you find in between sand particles and sediment grains. If you add in the alien species which have come in either on ships or been brought in and ballast water, and new records, because I found quite a lot of species which weren't recorded in the Plymouth Marine Fauna. Some may just have arrived recently, but some of them might not just been noticed before. You would end up with a pretty high number. I'm not sure what that would be, but we may find out a bit more during this Darwin Tree of Life program, which many people in the laboratory are involved in. We're not the only one who've done these sorts of studies. Other laboratories like the St Andrews one, which was the Gatti laboratory, which is actually one of the oldest laboratories, marine laboratories in Britain. It started just before PML. They get about 1,500 species, but they are on the North Sea coast, an open bay, so you wouldn't expect a high biodiversity. And I've just counted the fauna there, not the flora. And Isle of Man, the Port Erin laboratory, which is no longer in existence, they also did a marine fauna. And these books are really quite based on the Plymouth Marine Fauna, same sort of format. And they've got about 2,500 species recorded there, but they actually, they've taken in all the species for the Irish Sea. It's not just the Isle of Man. So I think this does indicate that Plymouth has got a really high biodiversity. I'm really retired, but I do some contract work for the Continuous Plankton Recorder group here in the laboratory. If you don't know the Continuous Plankton Recorder work, they tow equipment behind merchant ships on regular routes all around Europe and the North Atlantic, which collect plankton, and they analyse the samples that they bring back, and you can build up big distribution maps of the distribution of species and how they're changing over the years, because it's been done since for many years now. And that's why my other sort of role in the laboratory, apart from entertainment, is to do some work with educational people. I help out with Science Week when kids come in for a week during the spring, when different groups show them things. I show them plankton under microscopes. Kids love using microscopes and looking at stuff under them. And it's great to get the responses to see all the stuff there. Also help doing work experience, kids giving work jobs for them to do, to give them a bit of experience of working in laboratories. We do bio-blitzes, which many of you know. We go to particular sites, like we do one locally, which the last one we did was at Wembury. And we go there and we count all the marine species there and the plankton over a 24-hour period, but also all the other species. We get people who know about bats, know about moths, plants, and over the 24-hour period, you get the number of organisms that you find in that particular area. Also do talks to environmental groups, and this is a zooplankton identification course that I run in the MBA, a weekend 2-day introduction to zooplankton course. I usually do these other years and sometimes other courses. And you get quite a wide variety of people coming here from environmental companies, students, people who are interested, from other laboratories. I mean, this couple at the end here, they worked on a dolphin watching boat down in Cornwall, and they wanted to be able to speak to their clients about other things than just the dolphins. And so they came here and learned about plankton. In fact, they came 2 years running, which was pretty surprising. I even had a guy came from South Africa 1 year for a weekend course, which blew my mind a bit. But he stayed on a bit afterwards, and I spent some time with him, which I think he appreciated. But when you realise, when you do these courses for training people individually, that all the information, if you're looking at a wide range of zooplankton, it's widely spread through the literature in books and monographs, many of them out of date, expensive. Unless you've got resources like a wonderful library like we have here, it's very difficult to identify animals, especially if you're in isolation. So I got round that by I drew together a lot of information into 3 volumes of an identification guide to, it says marine zooplankton of southern Britain, but in fact it covers really the whole of Britain, the whole of Northern Europe. And these can be downloaded free from the Plymouth Sea website, which is the repository of all the sort of research that we do. You can get all the everyone that works here in the laboratory and past people, you can download the research there. Also ResearchGate, or it's on the National Marine Biological Laboratory, our laboratory's website, in the occasional publication series. And yeah, the 3 volumes, about 550 pages. It's quite a lot to download, but you don't need to do it. There's a good example here of the best bindings to use are black. If you use blue, they fall apart much quicker. And this is what it looks like inside. It's black and white, very traditional, with drawings of the animals, all the salient points that you should look at, various little parts dissected and drawn, and descriptions of the males, females, lengths, where you find them, further information. And I thought, at various times, it'd be quite nice to do a book in colour, with photographs of live drawing, mainly live organisms, live especially because they come out photographs so much better than preserved organisms, which go all opaque. And I was at a book launch here at the MBA one night, and I spoke to Julie and Mark Dando of Wild Nature Press, the publishers, and in a moment of wine-filled enthusiasm, I suggested that it might be worthwhile doing a book like this, and that is what I'm doing a lot of now. Usually in the mornings. I work in the lab, usually in the afternoons. And I need lots of plankton samples from that, and I get the sepia to get samples from L4 when they can, and this is them looking in towards the land. You know, this is 12 miles out from the lab. And we'll get open sea species here, and also at certain times of the year we get more oceanic species coming past. You never know what's going to turn up in the sample. It's always a bit of a treasure hunt. And they keep the samples cool, bring them in, and then I pick them up. Another place I go, you sometimes see me down the front here at the front of the Terrace Cafe, which is all closed up because of the COVID, but tables are all out now. And I go down to the pier here, right in front of the laboratory, and you'll see me pulling a net backwards and forwards along here, collecting a sample. And it's more inshore here inside the sound, so you get a lot of larvae of benthic species. But the tides bring in even some of the more oceanic species when they're around. I'm a bit stymied just now because a lot of the pier down the front was damaged during the winter, and it's all people working down here, so I can't get down. Another place I go is Bay Marina, and I can pull my net along some of the walkways here. It's a great place to go, but you can go any weather down there, even down on the Terrace Cafe. A lot of days there's waves breaking over the pier there. So I can come down here and take samples, and you're getting more benthic species and all the species associated with the sides of marinas, etc. Quite interesting. And this is the Spanish or maybe the French ferry behind. Another place I go is up in Ernesettle. And this is the River Tamar behind. This is the rail bridge which goes up the Tamar line up to Gunnis Lake. And this is a big inlet called Tamton Lake. And it's quite an interesting spot. It's the site of Bud's Head Mill. Bud's Head Mill the mill house was here. And what's interesting was it was a tidal powered mill. There's a big lagoon in behind here. At high water, the lagoon filled up through these openings here. All the mill's gone, of course. They filled up. Then when the tide was full, they dropped down the gates and closed the water. Then when at low tide, they would release the water to drive the mill. It was pretty innovative for its time in 1791 when this tidal powered mill was built, and it lasted till 1927. I had to put in the name of the builder, named Gonk. What a fantastic name. But I find that it's quite useful to me here. I get brackish water species. It extends the range of sort of species I get, and I can stream my net in the water as it comes in and out through the openings into the lagoon. I tend to use 100 micron net because that gets me small stuff and large stuff, and it doesn't get too much phytoplankton or sediment in it. So it gives me quite a nice range. Most people for routine surveys use 200 micron aperture nets, and that means a lot of stuff goes through it, but you've got to reach a compromise, and that's the sort of standard, and that's the size that MBA uses at L4 and also PML use too. Then I'll take the samples home wherever they are. And lately, of course, I've had to work at home. Originally, I was doing some of the photographic work in the lab. I had to buy a new computer, and I've bought another, I think, 3 microscopes. And this is one of them there. The sample is on a slide, just in a drop of water underneath the microscope. I've got my camera on a triangular head, connected up to a small television. And then when I've got the thing arranged and the way I like, I've got a remote which takes a picture, which helps reduce vibration. And this is a burrowing sea anemone larvae. There were a few of these around recently. That's a little polychaete worm and a Copperpod Norpolei. But I usually have things individually rather than several together. So I sometimes also culture organisms at home, like Marie Leboeuf used to do, just to be sure the thing I've got is what it's I think it is, from the eggs. And I've got a few things culturing down my basement just now, waiting for some eggs of copperpods to hatch. And I'll just show you some of the plankton we get. This is from L4, so these are species which live all the time in the plankton. This is Calanus, one of the best known copperpods. It's pretty large, about the size of a grain of rice, and it's used a lot in experiments because it's a nice large size. It's a very common species, but it's a very important species too. Most worms, the polychaete worms that we find, live in the sea bottom. Tomopteris is a pelagic one. This is a young stage. They have got paddle limbs which they swim with, so they are pelagic. Most people think of krill as an Antarctic species. We get them off Plymouth here. You get massive shoals in the North Sea. In the northern North Sea, I've seen the sea red with shoals of them. This is the northern krill Meganectiphines. Another species are the sea gooseberries or ctenophores, comb jellies. This is Pleurobrachia. It has rows of cilia fused together, and it beats them in Mexican waves. You can perhaps just almost make out the Mexican wave going down. And they're also iridescence. You get iridescence that happens. They're really quite beautiful to watch. Carnivorous species, Ketognath, called Sagitta, an arrowworm. And this is the sea butterfly, Limacina. Most mollusks, you can see it's a coiled shell, a mollusk. Most have a fleshy foot, which they slip along with, but this one is the foot modified and a couple of little wings, so it flies through the sea, flutters through the sea, so it's sea butterfly. And this is Podon, a little water flea. You may be more familiar with some of the species, Daphnia we get in freshwater. And it's got a big dark compi eye at the front, and you can see it's carrying young. There's the eye of some young ones in the brood pouch. And these are some of the more oceanic species we might go to L4. This is Mugia atlantica, a siphonophore, related to the Portuguese man of war. This used to just turn up late in the autumn, but this last year I've had it almost every month. This is a small jellyfish called Sulmaris corona. When we think of jellyfish, we usually think of the big lumps of jelly we see on the beach. In fact, these big jellyfish on the beach, we only have 6 species locally, whereas these hydromedusae, a slightly different group. Here in Plymouth, we have got 51 different species. This one is a really beautiful one. It's called Sulmaris corona, related to its like a sun shape. It's an interesting, it's an oceanic one. It doesn't have a bottom living stage. Most jellyfish have got a bottom living polyp stage. So they alternate between a bottom living stage, which it produces young ones, which go into the plankton, and then members in the plankton, which reproduce sexually, produce a young larvae, which goes to the bottom and then becomes a polyp. This one is a deep water species, so it can't reach the bottom easily. So its larvae just turns into another medusa. This is a large copperpod, a more warmer water one called Subucalmus crassus. It's a very large, chubby guy, and very feathery antennules front and swimming limbs. We get it occasionally. Last year it was reproducing. I was getting the young stages, the eggs, which it's got very unusual eggs. I was finding the eggs and also the young stages in the plankton. So it's just reproducing off here. These are rather beautiful. They're pelagic sea squirts called doleolids. They're related to the sea squirts that you get around if you saw John Bishop's talk earlier in the year, he spoke about invasive sea squirts that we find around Plymouth. The sea squirts have got, you find around in the marinas, have got an inlet siphon. They filter the water and then they put it out through an outlet siphon. And this one, it comes in one end, through the gills is filtered and goes through the other end. It's driven by cilia. These are muscle bands which allow it to move a bit faster. They're very beautiful, delicate looking organisms that turn up here most years. And this is Temera stylifera copperpod with a spiky bottom and very long furca, very characteristic. Turns up some years, but not always. But with global warming, we might find that more of these species are going to be spending more of their time here. And then there's all the temporary zooplankton that we find more inshore than at L4, but you got it at L4 too, because L4 is only 50 meters deep. And these are the stages of bottom living organisms. This one at the bottom is barnacle larvae. You can see it's got horns at the front, and this is characteristics of all barnacles. It turns into a later stage called a cyprid, which is the settlement stage. A polychaete worm, you can see a segmented worm with seta all over it and some eye spots at the front. They're really nice to watch in the plankton samples. Medusae called Abelia. As I mentioned earlier, there's 51 species of Medusae here in Plymouth, and there's about 140 in the general area around Plymouth, around the UK. I get this almost year round, this little Medusae. A lot of species are quite seasonal, but especially the jellyfish, but this little Medusae is still in the sample all winter. It has a bottom living stage, a hydroid colony, which produces these little jellyfish in the spring. And at the top here, we've got a sea urchin larvae, very unusual things, but very often larvae look nothing like their adult, but later on it'll develop a little spiky thing more reminiscent of a sea urchin here, and all these arms will be reabsorbed. This is a gastropod mollusk. It's got a little coiled shell, a couple of large lobes covered in cilia with which it feeds and swims. It'll grow and then finally settle out to live in the bottom. This is a really beautiful larvae, probably one of the most beautiful larvae in the plankton, and it belongs to a phylum called Pharonida. There are quite a few groups in the plankton that you won't be familiar with, and Pharonids are worm-like creatures. They're not worms, they're a phylum, all of them, as I said. Most of them are about 2 centimeters long, but some up to about 50 centimeters. They live in a tube on the seabed, and this is their larvae. And they have around the base, they've got a little row of cilia and all these long tentacles at the front. And they glide along really nicely. They look like something out of Kubrick's Space Odyssey. And usually when we have Science Week at the beginning of the year, we get lots of these young stages of jellyfish. We call it an Ephyra larvae. And it's the young stage of Aurelia, a jellyfish you're probably familiar with. They pulse beautifully, and they're great to look at under the microscope. So the Venki Marupali at the moment is actually culturing some of these. He wants to get some larvae from them to look at the cilia on them. This is a large accumulation of jellyfish. We get this from time to time, and of course it can be a bit of a problem if it's an area where there is tourism, they don't like it, and sometimes they're stinging jellyfish, and people are concerned about them sometimes. Some people think that there's been an increase in jellyfish blooming recently. Whether or not that's true, I don't know. They're just more reported now, and we get to know more about them. There's a sort of misconception that these blooms are due to sudden reproduction events causing these blooms, but in actual fact, they are produced early in the year, and that is all that is produced. They don't produce further the polyps in the bottom, and these accumulations are just due to tidal and current movements that they end up like this. In the autumn, these produce, they're male and females here, these produce a large little larvae which goes to the bottom and becomes a polyp, and most jellyfish die off. I can show you a quick, if you haven't ever looked down a microscope at zooplankton, here's a little video, and these are mainly copperpods here. This is Oithona, very common copperpod. This is Ditricarius anglicus. It's got little glassy eyes at the front, very square, boxy species. Little polychaete worm there with setae. This is a jellyfish pulsing away. You can see they've got a very mobile stomach moving along in the center. Lots of little copperpods jumping around, and also young stages of copperpods, like that one there. And also you get, because these are crustaceans, they molt, so they lose their exoskeleton and these end up in the plankton like that one there. These eventually get broken down and will be recycled. These are little shelled mollusk larvae or copperpods. A bundle of eggs there, it's fallen off some copperpods, I've been carrying them. Another Copperpod Norpolei there, Morithona. And this is a bit of a barnacle. Barnacles cast their skeletons as well. And all the casts get broken down and recycled again, as I say. Little hairy worm there with long seta on it. It's getting knocked around by things. It keeps closing up. Different sort of Copperpod Euterpina. And this is a fish egg of some sort. A lot of fish eggs in the sample just now. They lay quite earlier in the year. And that's a larvae of a sea anemone, early stage larvae. And that's Bidophia, a diatom called Bidophis sinensis, an alien species come from China. And this is another diatom called Coscinodiscus. I think it's Coscinodiscus wellsi, which came from America as an alien species. And apart from the normal plankton, we also get alien species here. They're mostly copperpods here, apart from this water flea. Acacia margalef is a copperpod which came from Spain. I first found it down in Millbay Docks, which funnily enough is where the Spanish ferry comes in, but it's also found up Severn Estuary and Southampton, and probably a lot of other places too. Euterpina americana was first recorded off Drake's Island in 1933, but it's never been recorded since. So about a year ago, I found some off the terrace cafe in front of the lab. And a couple of weeks ago, I was out at Pier ferries just along the coast, but that sometimes range a bit further than some of the local sites. And there was loads of them there. So I don't know why they've disappeared and suddenly come back. Pseudodaptus marinus is an Indo-Pacific species, which is spreading all around Europe. And about 10 years ago, it was found off the coast of Denmark. And subsequently, it was found in the continuous plankton recorder samples. It's really an inshore species, but the southern North Sea is really shallow, so it feels it's only about 3 inches deep, so it feels quite at home there. So it ranges quite far off the shore. So that's how the CPR survey will pick it up. A lot of these species are quite inshore, and as the CPR survey doesn't start till quite a few miles out at sea until it's safe to put the equipment over the side, they don't record many of these alien species. Acacia tonsa has actually been in the UK for many years. We'll get it up the Tamar River, dominating the population in some parts. And Pinellia avirostris, it's a water flea. It was first found in the CPR survey in the southern North Sea about 1999, I think. It wasn't known along the French coast going down towards Spain, but it was never in the Channel. And about 10 years ago, I found one at L4. And a couple of years ago, they turned up in great numbers off Plymouth. So they seem to be spreading and probably, I imagine, they'll become established. These copperpods and water fleas have something in common in that they lay resting eggs, which then go into the bottom muds, and then they can stay there for years even. And then when the conditions are right, they can hatch. So the population is maintained there. It's not, you're not always sure what effect these can have on other local animals. Some plankton, when it's been introduced in alien species, like some of the ctenophores into the Black Sea, started devouring all the local plankton and the larval fish, and they actually affected the fisheries there. They bloomed in such large numbers as species introduced from America. But it's more difficult to understand what happens, how these react with the local community. John Bishop and Chris Wood, they go around and check alien species in marinas like the sea squirts. They can go all around the UK, and it doesn't matter really what time of the year they go. The sea squirts will still be there. In the winter, they might be a bit scabby, but they'll always find them there. But these tend to have very finite breeding seasons. They'll only be there for a few weeks or months, and then they're gone. So you've got to choose your time to go places. And even Pseudoaptimus here in Plymouth, it seems to breed. I find females with eggs in the summer, yet the CPR survey feed finds them with eggs in the autumn in the southern North Sea. So we still don't know a lot about the distribution of these animals because there's not all that many sites around the UK which are collecting them. I then go on to my favourite animals. If I ever come back to work in marine science again, I'd had the chance, I would work in parasites. They've got such amazing life cycles. I'll start off with this one here, which you can probably recognise. It's like a woodlouse. It is an isopod related to little animals you get in your garden in damp places. And this one attaches to copperpods and feeds them on on the body fluids, and it hangs on there and molts through several stages, and then it eventually leaves the copperpod and moves on to a shrimp, attaches to the shrimp and feeds on it, and it feeds on it so much it stops it from breeding. And these isopod parasites can be a problem in shared shrimp farms abroad. This copperpod's got a double whammy. It seems to have a lot of protozoan parasites inside it and attached to it. This is also an isopod. It doesn't look like it. It's very fat, but it does go through a thinner phase. Now, I'll tell you, this is a little goby larvae that I got up at Ernest Settle. And it's got one on the nose, one behind the eye, one on the fin, and there's another thinner one here just on the base of the fin, and these feed on the body fluids of the goby. They get very fat, they drop off, go to the bottom, and they hide and molt, and they get very thin when they molt. Then they find another fish, they feed on it, get fat, they drop off again, they molt again on the bottom, they come back again. They do this several times, then they eventually turn into adults, produce young stages, which then starts the cycle all over again. This is a parasitic barnacle larvae. You can see it's a barnacle by the horns on the front. It's got quite a beautiful one. It's got this, they call it a flotation collar. I would say it was an impediment to swimming and floating. It's quite nice. It's got features in it almost like a fish scale. But this is a parasite of barnacles and crabs, and a later stage from this injects itself into the crabs, which forms a sort of almost like a fungus mycelium through the crab. It turns them into zombie crabs, and eventually emerges from the place where the female crab would lay her eggs. It's a great big ball of eggs, which is the female. And these hatch out, the eggs are released, and it turns into these larvae, which then start the process again. Of course, the crab can't breed. You'll quite often, if you ever pick up shore crabs and that, you'll quite often see parasites hanging out the egg flap. This is a caligoid copperpod. You'll know them best from places like salmon farms when they can be a big pest. They're really adapted for living on the surface of fish, flattened bodies, so they can stick on really close and are streamlined against water flow. Some of them attach also by a small tether at the front. This is a female with big sacs of eggs in behind. They cost millions of pounds a year to fish farms to control these. Recently, a lot of wrasse were being collected down here in the southwest and sent up to fish farms in Scotland, because wrasse are cleaner fish. They will feed on parasites on the surface of fish and dead skin, etc. And they were sent up and put into the cages with the salmon as a more natural control. I think that's probably stopped now because it was decimating the local wrasse populations. This is another copperpod, another very strange one. It's a parasite of tube worms and gastropods, benthic species, and it's the young stages which are the parasites. They're inside these organisms feeding on it, and then eventually an adult is released. This is a female with the eggs, and they reproduce, the male and females reproduce, and then the young Norpolei stages which emerge from these eggs then go and go back down and burrow into the tube worms or gastropods. In a normal copepod, this would have a whole range of feeding appendages along the front here. But because they're only for reproduction, they're totally missing these limbs. They're quite interesting species, but a few locally. I've actually got some females down below with eggs waiting to collect their Norpolei. And I find species I can't identify, which I think may even be new to science. You would think that you're not going to find any species locally which are new, but I don't believe it. Another interesting one are, I get a lot of in samples, the Cercaria larvae of trematode larvae. These are common parasites of fish. You get them in the guts and in the liver and place like this. It's got a little sucker at the front, sucker in the middle, and two dark pigmented eyes. It's got a Christmas tree tail, which it's a rather strange one. Some others have got a filled in tail, different species. It attaches to gelatinous plankton, usually, like this is a jellyfish. It's attached to, sometimes attaches to ctenophores, comb jellies, but also see it on chedognaths. This one has got over 100 on it. Now, this is going to be fed upon by fish, and it's going to be a big wham of infestation of parasites in one go. We also get in a lot of coppepods external epibionts of different sorts. This one here is, I forget what it is, Epibionts. And actually it's known to kill sometimes the host. It's a protozoan parasite. We get it occasionally. This is a copepod species, Parducita hebes. It's an oceanic species, it turns here occasionally. And I always get it with these little parasites attached to it, ciliate of some sort, I think. I never checked up what it is. Yet when I find it in the CPR samples, it doesn't have these. So some sort of problem to the more southern areas, perhaps. Another epibionts we get are these ciliates, very common on inshore species like these, Acacia. They've got long stalks, and when you get them live, you can see little cilia moving out there, and they're protozoan. And this Euterpina, this Eurytemera species here has also got similar ones on it. These, especially in brackish water, it seems to be quite a problem. I get populations of these, almost every one of them have got these things growing on them. It doesn't seem to affect some of them. I don't know whether they're actually feeding on them or just living on them. Commensals rather than parasites. It doesn't seem to affect this one because the female has still got eggs, but they could block up the pores and orifices, etc. So they could cause problems. So this Euterpina has got, they're green, so there must be little algae attached to it, as is this Ditricarius. It's got little algae attached to it too. And it's got eggs, so it can't be too affected. But it will possibly slow down the swimming. It will also make them larger and more visible to predators, possibly. And this is a species people will have heard me banging on about before, because I'm quite interested and would like people to know, hear more about it and to recognize them, because I'm sure over many, many years they've been misidentified as Copperpod Norpolei. They are a crustacean very closely related to barnacles. Barnacles are in infraclass Cirripedia. These are in infraclass Facetotecta. They differ from barnacles among some things, but they don't have horns on front like barnacles have. They're called Facetotecta because they have facets like a diamond, and these are the facets here. This is a cast, this is a molt. Like all crustaceans, they molt, so you get these empty casts after they've molted, and little facets like a diamond. They're pretty beautiful little animals. They molt several times, and then they turn into this cyprid stage. And then this cyprid stage will go and settle like barnacles do. And they are believed to be parasites, and they go and parasite something. But after 125 years, we don't know what they parasitized. And that seems pretty impossible, but they're not very numerous, they're very small, and they just disappear. They're a lump in something. I don't really know what, but something very small. I've been, they're found in all the oceans, in small numbers. Some are much larger than the ones I get here. I get about 3 species. 2 of them may have never been described. And there's a place in Japan, I think they've got about 40 different species. And so they may be quite important, but we just don't know. I've been trying I wanted to try some biochemical methods to try and identify them, but it's been sort of stopped while this COVID problem was on the go. So I haven't got around to it yet. And this is the last slide, which is payback time. This is a parasitic copy. It's not a very good picture, but it's got a parasite attached to it. So it's a parasite by another parasite, and we call that a hyperparasite. Thank you very much.